This photograph shows a melanoma lesion. Its asymmetrical, irregular borders represent a highly aggressive skin cancer that quickly penetrates deep into tissue. For a stage 2B to stage 4 diagnosis, the primary treatment is surgical removal. But even after a successful surgery, a profound fear remains. Microscopic cancer cells can escape the primary tumor site, silently circulating through the body and waiting to metastasize in other organs. On August 19, 2026, the medical community received clinical data showing this exact threat could be actively tracked and eradicated. Pharmaceutical companies Moderna and Merck released the results of a joint phase 3 trial that demonstrated a viable, highly effective method for treating this terminal diagnosis. The data released that day provided the first phase 3 evidence that a personalized mRNA approach could outperform the standard immunotherapy used to treat high-risk skin cancer. The data came from the INTerpath-001 phase 3 trial. Researchers tested an experimental mRNA vaccine called intismeran and paired it with an existing immunotherapy drug known as Keytruda. This chart compares recurrence-free survival. Patients on combination therapy experienced a 44% reduction in recurrence risk compared to Keytruda alone. Updating to distant metastasis-free survival, the treatment stops the cancer spread. The gap widens significantly over the 18-month mark. These specific trial metrics prove the treatment actively prevents the cancer from re-emerging in the body, rather than just managing the symptoms of a recurrence. To achieve this, intismeran relies on the same underlying mRNA technology that gained global prominence during the COVID-19 pandemic. But instead of a generic, mass-produced formula, this vaccine requires a patient's unique genetic fingerprint. Scientists take the specific tumor removed during surgery and sequence its DNA to map the exact mutations driving that individual's cancer. This DNA map is translated into a custom mRNA strand encoding 34 patient-specific mutated proteins called neoantigens. Once administered, the mRNA enters the patient's cells, generating these markers. This instructs the immune system to recognize those exact proteins and lock on to any remaining cancer cells. Because of this, the patient's own immune system becomes a highly precise assassin, hunting down an individualized biological target. The release of this clinical mechanism and the phase 3 data triggered an immediate reaction across global financial markets. Tracking Moderna's stock price on August 19, 2026, shares surged nearly 117%, peaking above 41 reals in a single session. Scaling out to total market capitalization, the valuation rapidly expands over 130 billion reals, a massive recovery from the year prior. Institutional investors injected huge sums of capital into the firm, betting heavily that this targeted mRNA approach is a viable, scalable commercial reality. If this biological mechanism can successfully intercept and defeat melanoma, researchers are now asking if it can be applied to other solid tumors. The INTerpath clinical program is already answering that question. Merck and Moderna have expanded their trials to test this personalized mRNA approach against lung, bladder, and kidney cancers. This table outlines the current and future challenges to widespread vaccination in cancer patients. Creating a custom drug for every single patient presents massive hurdles in the scalability of manufacturing, as well as significant regulatory complexity for health agencies. Overcoming those logistical bottlenecks will take time and capital, but the underlying biological proof of concept holds up. Oncology is actively moving away from purely reactive treatments and toward proactive immunoprevention. Behind the phase 3 clinical data and the billions in market capitalization are individual patients and families who previously had no viable options. Pushing an unproven technology through the clinical pipeline required sustained corporate vision and substantial financial risk from leadership. A late-stage melanoma diagnosis historically offered very little hope. Now, by identifying the exact genetic signature of a tumor, patients have a realistic path to walking out of the hospital completely cancer-free. The success of this trial validates the massive investments into mRNA research, moving it from a theoretical breakthrough to a tangible treatment option for patients with resected melanoma. While the technology is still being tested for other solid tumors, the results in melanoma suggest a shift in the oncology timeline, where high-risk patients have a measurable biological defense against the return of their disease.